Welcome to our very last research insider conducted by MBIOS for the term 22-23. My name is Yi Jie and I'm today's moderator. So just a little housekeeping before we kickstart our session. As you know, we are broadcasting our session live on Facebook. So do share this live stream with your families and friends who are interested in knowing more about today's topic. If you have any questions, feel free to type in the chat box or raise your questions during the Q&A session. Introducing our gold-tier sponsor, UKEC. UKEC is a leading UK education consulting organization with over 20 years of experience working closely with UK universities. UKEC is a reliable source of advice and assistance for a vast network of international students who wish to pursue higher education in the UK. Now, I would like to share something exciting with the participants before we kickstart our session. MBIOS has launched a program called MBIOS Certificate. Only MBIOS members are able to claim this certificate, which can serve as an addition to your CV and stand out from other applicants. It works as an accumulation. For example, you have joined 3 events and you get a bronze certificate. And later on, you have joined 2 more events and 3 plus 2 equals to 5 events in total, which means you are eligible to get a silver certificate. If you wish to learn more about it, feel free to visit our social media. First and foremost, I would like to introduce our speaker for today's session, Mr. Yi Zhe Koh. Also, he is the director of the postgraduate department of MBIOS. Let's welcome him. Mr. Yi Zhe Koh is an esteemed PhD student at the University of Bristol School of Physiology and Pharmacology. He's a recipient of multiple magnificent awards, like the University of Bristol International PhD Studentship and the J.F. Mitchell and P.J. Roberts Prize. Mr. Yi Zhe Koh is going to share with us all the research that was used to accomplish such a feat, from pluripotent stem cells to heart muscle cells. Additionally, he's eager to go into greater detail about the investigation of heart tissues and explore the current limitations of some heart disease treatment methods. Without further ado, I would like to invite our speaker, Mr. Yi Zhe Koh, to share his knowledge. The floor is all yours now, and you may share the screen. Thank you, Yi Zhe.
Hi, everyone. Can you see my screen all right?
Yes, we can see.
Okay, thank you very much for the introduction, Xinjie, and a very good afternoon to everyone. My name is Yi Zhe, and I'm currently a second-year PhD student at the University of Bristol, and I'm also the current postgraduate director at Malaysian Bioscience Scholars. It is with great pleasure for me to be talking to all of you here today. And in today's session, I aim to introduce to you the bulk of my PhD research, which is on electrophysiology and also on stem cells-derived cardiomyocytes, which are essentially our heart muscles. And towards the end of the session, I would also like to give a bit of an insight into the PhD application process in the UK. So I completed my A-levels in 2017 in Sunway College, and you will have noticed that I didn't actually take biology to pursue a degree in actuarial science. And obviously that didn't work out. I happened to meet a very inspiring chemistry tutor back in A-levels, and he was the first one who introduced me to the concept of pharmacology. And after some time considering and deciding, I have decided to do an undergraduate degree in pharmacology. And it was a 4-year Masters of Science program, with my 3rd year spent doing a year of industrial placement. That was back in 2020. It was during COVID time, so I was back in Malaysia, and I did my 1-year placement in a Malaysian-based pharmaceutical company called Hobbit. So some of you here who have majored in analytical chemistry or pharmaceutical sciences may be quite familiar with this fancy-looking machine here. So this is a HPLC machine, a high performance liquid chromatography machine. So it is used to separate components from a drug mixture. So my project at the time was to develop a capsule formulation for a drug called dutasteride. It was a very interesting project. I also had a chance to work on a 2-month internship at the Tropical Infectious Disease Research Unit at University Malaya. My project back then was to isolate salmonella from egg samples, which I collected from the local supermarkets. And you could see that I gave a thumbs up here because I did manage to isolate some salmonella from these egg samples, which I shouldn't be too happy of, because if someone actually consumed these egg samples, they would be at risk of a salmonella infection, right? But I was pretty excited because my experiment protocol has worked. And this is me wearing the graduation gown. So I've graduated last year in 2022, July 2022. And here's me holding my master's thesis. To stay on to do a PhD at University of Bristol, specializing in stem cells derived cardiomyocytes. So I would like to give an introduction to electrophysiology. Some of you here who are from neuroscience or maybe human physiology background may have come across this term. So how many of you here have come across this beeping machine? Quite a few. So this beeping machine here actually shows the patient's heart electrical activity. So what the clinician would do is that they would place a few electrodes attached onto the patient's skin, and then these electrodes will be able to pick up electrical signals produced by the patient's heart each time it beats. And these are called electrocardiogram. A clinician will be able to study the trace and be able to tell the patient's heart rate. And also if the patient has an abnormal heart rhythm, such as a ventricular tachycardia or atrial fibrillation, the doctor will be able to tell by looking at these traces. So you may be wondering, where does the electrical current comes from in the cell? And now I would like to introduce you to the concept of ion channels. So in our body, there are many different types of ions. There are positively charged and negatively charged ions. So in this context, there are two very important positively charged ions. One is the sodium ion, and the other one is the potassium ion. And when there is no external stimulus, when the cell is at a resting state, these ions would always maintain a fixed concentration gradient. And what I mean by this is that there will always be more sodium ions outside the cell compared to inside the cell. And there will always be more potassium ions inside the cell compared to outside the cell. And they are separated by this lipid bilayer membrane. So this cell membrane acts as a wall, and it will not allow ions to pass through in or out the cell. And the only way an ion can pass through to flow into the cell or out of the cell is through something called the ion channels. So the ion channels are a group of protein embedded on the cell membrane. And when they are open, these ions could flow through these channels and go into the cell. And there are various types of stimulus that can activate these ion channels. So that depends on the type of ion channels we're talking about. For example, the voltage can receive an electrical signal. These channels would open and then allow ions to pass through. Another type of ion channel is called the ligand-gated channel. So these ion channels will respond to small molecules such as neurotransmitter. So this neurotransmitter, like adrenaline, they will bind to certain types of ion channel. The channel will open and allow ions to pass through into the cell or out of the cell. So in this example, we have sodium ions flowing into the cell according to their concentration gradient, because there is more sodium ions outside of the cell compared to inside the cell. So they will flow into the cell through these ion channels. So we say there is a net movement of positive charges flowing into the cell. And that means there is a net movement of current flowing into the cell. And as more positive charges flow into the cell, there are more positive charges built up inside this cell membrane. So the membrane voltage will become more and more positive. And this process is what we call depolarization. And the opposite of depolarization is repolarization, is when the cell membrane becomes more and more negative. So you can imagine there are many different types of ion and different ion channels in a heart muscle. The three most important ions are the sodium ions, potassium ions, and calcium ions. And it is the time-dependent movements between these different ions that creates this sequence of changes in membrane voltage. So for example, at phase 0 here, we have the voltage-gated sodium channels opening, sodium ions flow into the cell, which then cause the upstroke here that we are seeing. So the cell becomes more and more positive. And then we have the potassium ion channel open, which cause this, what we call a transient outward current. And then the calcium current comes into the cell, which causes this plateau phase. And finally, the repolarization phase is caused by a few different potassium ion channels. So it is this group of different ion channels working together, which then causes this very beautiful sequence of change in membrane voltage, which then give rise to this shape, which scientists call it the action potential. So the cardiac cells or the heart cells uses this electrical signal, this action potential, to communicate with one another and coordinate a contraction. So when an electrical signal arrives at the cell, it would open up a type of voltage-gated channel called the L-type calcium channel. So when this calcium channel open up, it would allow calcium ions to flow into the cell. Okay. And these calcium ions would then bind to a type of receptor called the ryanodine receptor. This ryanodine receptor will then open up something called the sarcoplasmic reticulum. So this sarcoplasmic reticulum here, you can think of it like a storage room filled with calcium ions. So when this ryanodine receptor opens up this sarcoplasmic reticulum, it will allow even more calcium ions to flow out of it and fill up the entire cell. And when there is sufficient amount of calcium ions filling up the entire cell, these calcium ions will start to bind onto a type of protein called troponin C, which lies on the actin filament. So these myofilaments are the units in the cell that are responsible for cellular contraction. So this will bind to the troponin C, and this actin and myosin filament will start to slide alongside one another and hence initiate a contraction. So this whole process is what we call an excitation-contraction coupling. Excitation means electrical signal arrive at the cell, which then leads to a contraction. And there is a way which we could study the contraction in these cells, that is studying the electrical properties of these cells. And a very common technique that we could use to study the electrical signals in these cells is a technique called patch clamp. And here is a common setup of a patch clamp experiment in the lab. So this is the stage filled with bath solution, and the cells are in the bath solution. There is usually an inverted microscope here to allow the researcher to identify where the cells are. This perfusion tube here is important to regulate the temperature of the bath solution. It has to be always at 37 degrees Celsius, at the physiological temperature. And here we have the recording electrode in a glass pipette. And this is the pipette that will be coming into contact with the cell. And we will record the ionic movement of the cell, which then feeds into an amplifier, a digitalizer, and into a monitor, which scientists can then view the electrical signals. So this is the setup in my lab. You could see this is the bath solution. In the middle of the bath solution, there is this round glass cover slips, which we put the cells on. Underneath the cells are the inverted microscope. We have the perfusion tube here to regulate the temperature of the bath solution. This is a stimulating pipette, which we use to inject current to the cell to basically stimulate the cells. And this is the recording pipette that will be coming into contact with the cell. And this is a 40 times view of the recording pipette coming into contact with a single cell. Okay, so this is the general procedure researcher will use to patch a cell. So first, we will look for a healthy looking cell. these are typically cells that are round and are more sort of 3D-like that we think is good to patch. The researcher will then slowly move the pipette down until it just touches the cell. And when it touches the cell, it will form something called the gigaohm seal. So this is a very, very tight contact between the pipette and the cell membrane. It is called a gigaohm seal because the electrical resistance between the pipette and the cell membrane is literally more than a gigaohm. And also because the shape of this cell membrane actually resembles the symbol of ohm. So it is a very tight contact between the pipette and the cell membrane. And this is also to ensure that there is no current leaking out from the pipette. And now we can apply a mouth suction to break the cell membrane so that the pipette has electrical access to the inside of the cell. And we call this the whole cell configuration. So what the scientists would do is normally we would have an external syringe connected to this pipette, and then we can apply a slight negative pressure with the syringe, which would then break the cell membrane. And this is how a typical cardiac action potential would look like. So by studying the shape of this action potential, we will be able to study the function of different ion channels. So for example, if a patient has mutation in one of the potassium channels, we would expect the phase 3 of the action potential will be slightly prolonged. Then we would know something is wrong with the potassium channels in these patients. And different phases depicts the movement of different ion channels. And now I would like to introduce you to the experimental model that I use in the lab. So these are stem cells derived cardiomyocytes, essentially heart muscles that I culture in the lab. And you can see that they're spontaneously beating under the microscope. So some of you here may not be too familiar with the concept of induced pluripotent stem cells. So I will just generally brief through how we get these cells. First, a clinician would obtain a skin fibroblast from a patient's skin via a procedure called biopsy punch. So what the clinician would do is normally they would have a puncher. They will punch a tiny little hole from the patient's skin. So this would collect like a tiny cluster of skin cells from the patient. They will culture them in the lab. And then they could reprogram these skin cells into a pluripotent state. So what I mean by pluripotent state, so pluripotent means that the cell is back to an embryonic state, which allows them to turn into any type of cells we want it to be by exposing them to the right environment. So by introducing a group of proteins called the transcription factors, we could reprogram these somatic cells, these skin cells, back into a pluripotent state. And once they are back in this pluripotent state, we could differentiate these cells into cardiomyocytes or into any cell types we want it to be. And depending on the cell type we want it to be, we need to expose these cells to the right environment by adding in the right amount of growth factors, proteins, small molecules, and all sorts of stuff. So in this case, we are interested to differentiate the cells into heart muscles. And I can also modify the differentiation protocol to further purify the cells into chamber-specific cells, such as the ventricular cell or the atrial cell. Before induced pluripotent stem cells were introduced by a Japanese called Shinya Yamanaka back in 2006, the only way we could get hold of these stem cells is through an actual embryo. So this is an early stage embryo. So this whole thing is called a blastocyst. It's a very early stage embryo. The outer layer of the blastocyst would then later become the placenta, and the inner mass of this blastocyst would eventually become the embryo. But if we isolate the cells in the middle here, this would actually become the embryonic stem cells. And these are the stem cells that are pluripotent, that could turn into different types of specialized cells. But for obvious ethical reasons, we cannot just destroy embryos for the sake of experiments, right? So this induced pluripotent stem cells is a real game changer because we don't have to destroy embryos for stem cells. We could now just extract a tiny little skin cells from the patient and then turn them into pluripotent stem cells. and expand them as much as we would like it to be. And then we could differentiate them into any cell types we want them to be. And this is done by introducing 4 different transcription factors. So transcription factors are a group of proteins that binds to a cell's DNA, and they could then upregulate or downregulate certain genes. So upregulate means they would increase the expression of some genes, or downregulate means they would reduce the expression of some genes. So in this case, we are interested in genes that are related to pluripotency. So these 4 transcription factors act to increase the expression that are related to pluripotency. And the scientists could introduce these transcription factors by using virus as a carrier. So they infect these cells with viruses. This virus carry these genes that are related to pluripotency. And then once the cells are infected with virus, the virus will inject these genes into the DNA, and then these cells would then produce transcription factors and upregulate genes that are related to pluripotency. And this is what we call induced pluripotent stem cells. It is a further 2 weeks process. So after we get our induced pluripotent stem cells, we need to go through another 2 weeks differentiation protocol to make them into stem cells derived cardiomyocytes. So by around day 12, we will start seeing beating heart cells. And that is how we know the differentiation protocol has worked successfully. We would also need to validate that the cells are actually cardiomyocyte, and this is done by staining these cells with certain antibodies that are specific to proteins that are only expressed in cardiomyocytes. If you are interested in how we differentiate these cells into cardiomyocytes, you could read it up online in the literature. It basically targets a signaling pathway called the Wnt signaling pathway, WNT. So yeah, we expose these cells to certain growth factors and small molecules that all targets this Wnt signaling pathway. And after 14 days, the stem cells will become cardiomyocytes. So what are some application of these cells? Why is it important for us to study these cells? Well, the first obvious one is the development of personalized medicine. So as I mentioned earlier, these cells are extracted from patients' skin. So you would expect the stem cells would have the exact genetic component as the patient. So for example, if the patient has a certain rare mutation, then we would expect the stem cells to have the exact same mutation at the same location. Okay, then we could use this cell to study the mutation of the patient, and then we could tailor some therapy or some drugs that are specific to treat this patient's mutation. And that is what we call personalized medicine. Another one is cardiac cell transplant. So, well, for obvious reason, we cannot ask someone to donate their heart, right? Well, maybe it could be possible, but on very, very rare occasions. But with stem cells, heart transplant could become a possibility in the near future. And currently there are research that is going on. So these are stem cell plasters in which we coat a layer of stem cells onto these patches. And then we can stick on these patches onto damaged part of the heart in hopes that this plaster, the stem cells on this plaster would regrow the vessels and repair the damaged part of the heart. So these are called stem cell plasters. Another application is cardiotoxicity assay. So before every drug reaches the market, they need to be tested for their potential side effects. And cardiotoxicity means testing if the drug has any side effect on the heart. And we could do that by using stem cell derived cardiomyocyte as an experimental model to test the drug's effect on the heart in the lab. Lastly, we could use these cells to study congenital heart diseases in the lab. But despite all of this very interesting application, there is currently a big limitation that is stopping us to use these cells clinically. So these stem cells derived heart muscles, they more closely resemble a fetal heart cells, and they do not correctly reflect an adult heart condition. And one example of this is the structure, the shape of these cells. So stem cells derived cardiomyocytes, they are more sort of irregularly shaped. they have these very disorganized myofibrils. So these myofibrils are the contractile element that is responsible for contraction. So you can see that they are very disorganized compared to adult humans cardiomyocytes. These are much more organized. There's this very well-aligned striations. So you could imagine that this would have implication on how the stem cells contract as compared to how an actual adult human cardiomyocyte would contract. So one of the main focus of my PhD is to try to find ways in which we can mature these stem cells derived cardiomyocytes so that they more closely resemble the shape, the properties of an adult human cardiomyocyte, so that we could use these stem cells to better model adult heart diseases. And lastly, I would like to talk a bit about my personal experience applying to PhD in the UK. So I can only speak for UK universities because I personally haven't applied to any other countries. This is the general timeline of a PhD application in the UK. So you will first start out by having an informal chat with the potential supervisor. So this is probably optional, but it's always a good idea to approach the potential supervisor before you send in a formal application. This is to make sure that their lab is open to having new students, and also you could make sure that your research interests align and really to gauge whether the supervisor is happy to support your application. So after that, you could send in a formal application, and usually a PhD application will require these few documents. The first obvious one is a personal statement, a research proposal. So in my case, the research proposal is quite a simple one. It was a one-page document to outline the objective of my PhD project and also some research techniques that I plan to implement during my PhD. And this is just to make sure that the university have all the facilities that could help me reach my research goals. Academic transcript, a resume, and at least 2 recommendation letters from your undergraduate tutors. So if you're shortlisted, you would have a formal interview with your supervisors. So during this formal interview, you could be asked to make a presentation. It could be something you have done during your undergraduate study, during your final year research project, or it could be something you have done during your internship. If not, you could be also tested on your basic knowledge on the field that you're applying for. And depending whether you're applying for a scholarship or research fundings, that would be another round of interview. Or if not an interview, there will be a separate screening process by the scholarship committee. So I would say this is the most competitive stage throughout the entire PhD application process. And it is mostly based on academic merit. So you need to have an excellent academic track record. And if you're successful, then you will get your offer. I would also suggest a very good platform to start your search for PhD projects. So this is the website, findaphd.com. They have a function there in which you can like filter a list of PhD project that comes with funding. And it is also very important that you always discuss with the potential supervisor whether the fundings are available for international students, because a lot of fundings are only available for local students in the UK. So it is, again, I think it's a good idea to approach the potential supervisor before you send in the application. And most importantly, to ask them if there are fundings available for you. So this is the more traditional type of PhD in which the funding is attached to a specific project that you're applying for. If not, you could also consider a doctoral training program. So doctoral training programs are sort of more structured PhD programs. They are usually 4 years long. And during the first year of your PhD, you will be doing lab rotations. So lab rotations are like, you will be doing a few separate research projects at different labs. And you will also be going for a few lectures. So that is during your first year, and you will move on to your main PhD project during your second year. And there will be multiple checkpoints along the way. So you might have to attend conferences or presentations organized by the funding body throughout the year. So it is sort of more structured to make sure you are going in the right direction. And they are also usually very well funded. But I heard the quota for international students is quite limited. So maybe they will only have 30% of the places reserved for international students. So it's a bit more competitive. But some of these funding bodies, for example, the GW4 is a collaboration between 4 different universities in the southwest of England. So it is a collaboration between Bristol, Bath, Cardiff, and Exeter. So there are some PhD projects there which would allow you to like move around different labs and move around different unis. So it might be quite exciting for someone who likes to travel, and definitely just keep your options open and look out for these doctoral training programs as well. And one thing I would like to also emphasize is that there's a very common misconception that you would need to have a lot of research experience. You will need to have understand all the publications before you could apply for a PhD. But based on my own experience, that is not necessarily the case. You don't need to have a lot of research experience. You're not expected to know everything before you start your PhD. But most importantly, you need to demonstrate passion for the subject. You need to clearly state in your personal statement or during your interview, why do you want to do a PhD and why do you want to pursue research in this specific field? And also, definitely you need to have an excellent academic track record to be competitive, especially in terms of applying for scholarships and research fundings. And that is all I want to share today. I'm happy to answer any questions if you have any, or else I'm also happy to take this offline. You could message me or send me an email after this session is over.